It's my great pleasure to introduce Dr. Geoff Wells. He's from UCL, School of Pharmacy in London. And he's going to tell us a bit about what you might want to think about after you've done your docking. And I just want to say a couple of things about why I'm so happy that Geoff was available to talk to us all today. So Geoff is a really amazing example of somebody who is predominantly an experimentalist. So Geoff is a medicinal chemist, but he uses molecular visualization and modeling throughout his research just to help him think about things, to explain things to other people, and to generally guide his experiments. So I think it's extremely helpful to have people using our models from that perspective. And it's very important to have members of the experimental community using our tools and interacting with us, because that's actually, I would argue, how we build better models. And I have to say that most of the modeling projects I've been most proud of have often been done with experimentalists who have run their own models, because it's actually the experimentalists who really understand very often their system, and it's understanding the system that is key to building a big model, a good model, rather than just understanding the modeling. Actually, understanding the modeling is the easy bit, it's the system that's hard. So if it's okay, I'm now going to hand over to you, Geoff. So we're very excited to hear your talk. Thank you very, very much.
Okay, fantastic. Good afternoon, everyone, and welcome back. It has been a long day, I'm sure, and I will keep an eye on the time and also be fairly light touch, I think, in terms of what I'm trying to say to you. Hopefully you can all see my screen. Sarah, can you nod? Good, excellent. So as Sarah mentioned, I'm a medicinal chemist, and it's really nice to get the opportunity to talk to you today from slightly looking in from the other side of the research community into what docking can do for people who run experiments such as myself. So I do some medicinal chemistry. We also do some ligand binding assays, and I'll try and give a bit of perspective on the kinds of things that we look for, and also some health warnings on the things I say. The first thing I'd like to share with you is a quote, and it is something that will offend some of the chemists out there. So this is Frederick Soddy. He's a was a radiochemist, worked with Rutherford on radioactivity experiments. And he said that chemistry is termed by physicists as the messy part of physics. I think some chemists might not like being described as part of physics, but he noted that physicists should not be permitted to make a mess of chemistry just by evading it and pretending it doesn't exist. So we do experiments, and I guess once you've carried out a docking study and tried to process its results, then you will inevitably at some point want a chemist to make some of those molecules or make some derivatives of things you can buy. So that's where we come in. So we do some lab-based experiments to initially make things and then begin to test them. That's our goal. But there's a health warning on what I'm going to say to you. So as a medicinal chemist, I appreciate, because I've read it in the literature, that we differ in our opinions on things. So don't necessarily believe everything that I tell you, because medicinal chemists don't always agree, except, as it turns out, about what they don't like. So this paper from 2012 set a series of medicinal chemists kind of against each other in a competition to identify promising fragments from a drug screening set. And they all came up with different answers. And they only tended to agree about what they disliked in some of the hit compounds they were asked to develop further. And they only used one or two criteria when selecting hit molecules to work on. So I guess what you can do as molecular modelers, people who carry out docking studies, is consider how you might inform chemists, and how to give them more things, more than one or two things to think about when choosing molecules to develop and pursue in experiments. So let's just recap on why we're doing this first of all. Well, invariably, if we're doing docking studies, our goal is to develop a new ligand, perhaps an inhibitor for a target protein for some kind of therapeutic purpose. That's what we normally want to achieve, or perhaps to use in some kind of chemical biology application to understand the biological process by intervening in that process with a small molecule. And often for new targets, new biological processes, there aren't ligands available that we can already use, so we need to design a new one. And where are we working? Well, we would like to identify a ligand binding site, and of course you've seen approaches to identify ligand binding pockets. There is software available out there that you can use, fpocket, ftmap, for example. You can use molecular dynamics to identify cryptic pockets in proteins, and you can think about interacting molecules with protein-protein interfaces, etc. So you have a bewildering choice of potential sites on macromolecules that you can bind drugs to. And how? Well, our design approaches are many and various. We can look at known substrates of proteins, for example, or known inhibitors. We can dock those, we can try and modify those, and we can adopt completely ab initio design approaches. Some of those will be modeling based, some screening based. But inevitably, from our perspective, we're interested in how molecular docking might play a role and how docking small molecules to proteins might play a role. And we can find our ligands in various databases. This is the zinc database, which is one of the more commonly used repositories of small molecule structures. Contains many millions of molecules, and you can select subsets of those molecules to use in your docking studies. For example, FDA approved drugs or fragment type drugs or everything that's for sale, and you can download all of those if you have the bandwidth to use in your docking studies. So at some point we have to make a choice about the molecules we dock and how we process those before we start. And that's kind of important because, as you've heard over the last few days, parameterizing proteins is important. So understanding that PDB structures might not be perfect is a significant issue in molecular modeling. And also we need to appreciate when simulations can go wrong. So you've heard something about that over the past few days as well. And I guess you may not be too worried about the little guy in all of this, the small molecule ligand that you're working with. You might think that's the simple part, but that can go wrong too. And even in the PDB, there are some examples of bad ligands. So this paper from 2012 by the Cambridge Crystallographic Database highlighted some of the issues that can crop up with ligands in crystal structures. So this example is what looks to be a kinase inhibitor. And you can see here that we have a rather odd bond angle between the pyridine ring, which is flat, and its substituent, which should be in the same plane as that pyridine ring. That's this part of the molecule here. And that is a bad bond angle in the PDB. But you can also run into bad bond lengths and clashes and such like. So be a little bit careful with the things you use as your benchmark from the PDB database. It's just a bit of a health warning about where we start. Now, that kind of means that when we build our molecules from databases, often it's easier to download large numbers of structures in a 2D format because the file size is smaller. You can even download them as kind of one-dimensional smile strings. And that means we need to process them to make 3D structures and add in atom types and partial charges and assign a pKa to those molecules as well. So if we were to start with a molecule such as the one shown on the slide, this is propranolol. we would use a tool to convert it into a 3D format. Now, there are various commercial pieces of software that will do this for you within Schrödinger and MOE, etc. But MolConvert is a freely available one. That's probably the best one that I've used. OpenBabel will do this, as will Balloon and some other pieces of software around. Some are not so good. Some cause problems and introduce errors. So you need to be quite careful with your choice of tool here. I'll put the web links to some of those pieces of software at the bottom of this page. So we take our 2D structure, we make it 3D, and the things we might want to watch out for are bond geometries and stereochemistry. So propranolol has a chiral center. We need to check that that's been introduced in the 3D form. And if we don't know the stereochemistry of a particular site, we might want to make both forms of the stereo center. And of course, if you've got more than one stereo center, that leads to many different versions of that particular starting structure. And the other thing we need to do is check that the protonation state of the molecule is correct. So in this case, propranolol contains a secondary amine, a physiological pH that's likely to be protonated, and that amine group would carry a positive charge. So we need to check that we've got those things right. And we need to have some faith in our software that does this, because it's very difficult to go through large sets of molecules and check that it's right all the way through. Sometimes you only see this once you've got your docking results. So let's take a look at two broad approaches from a ligand perspective that can be used in drug development approaches. So I guess something that's cropped up quite recently, and largely because of coronavirus, is the potential of repurposing. It's something that's been attractive to pharmaceutical companies all the way along, I guess, because it provides a potentially quick route to market for a new product. So if you can dock a known drug to your protein, if it binds well, if it has a high affinity, it presents another use for an existing medicine. Now, those medicines are already formulated, they have known safety profile, clinical trials have been conducted. We know things about their side effects, their drug interactions, their contraindications and cautions, so that's all good. If you're financially minded, you may be a bit worried about your intellectual property position. And I guess one of the most common hurdles you run up against with repurposing approaches is that the clinically used drugs that are already licensed are often not a good match for a new target. So it may be just that the chemical space isn't right for your particular target. And we'll take a quick look at fragment-based approach as well. And this is a prevalent route to new drug design. It's used in the context of high throughput crystallography, and I'll show you a little example of that a little bit later, but you can also use this kind of approach for docking type studies as well. Now, obviously this is drug design starting at the very beginning, so it's time consuming. Fragment-based approaches are a little bit faster than traditional approaches maybe, but it's still expensive, and all of the preclinical and clinical steps need to be completed, and a new drug application needs to be made at the end of a drug design process. But you have the potential to come up with a new bespoke molecule that is a good fit for a particular target. So there are pros and cons to both of those kind of two extreme approaches. So let's take a look at FDA approved drugs or compounds that are approved for administration to humans, I guess, to cover more bases. Now, if we're doing a docking type study using this range of molecules, then we need to be mindful of a few things in the output. Now, often we're interested in protein targets that are within the body, within our human cells. And it's not necessarily the case that all of the drugs in the database have access to those tissues. For example, some drugs are used exclusively in creams for the skin, or ointments or gels. So they're applied peripherally. They may have properties in terms of lipophilicity that makes them more amenable to penetrating the skin rather than other biological membranes. They may be toxic if you administer them systemically. So we need to bear in mind that some of the compounds that crop up in our docking hits that we might be interested in may not be safe medicines. The same is true of drugs that are just applied to mucosal tissues, for example, applied to the nose or sublingually, things that you may not fully ingest. We need to bear in mind that they may not be safe for systemic administration. So that may limit the pool of drugs we can use within that FDA approved set. And some of those drugs have human targets already. That may be a good thing or a bad thing. There's a potential for toxicity there. Some small molecules interact with pathogens. So here where we have about 215 small molecules that target pathogen proteins. And I guess here we're looking largely at antibiotics and antivirals and antifungals of various kinds. And some drugs interact with non-protein targets. So here we'd be thinking about DNA and RNA in particular. And of course, there's a broader group of approved compounds, which may be diagnostic agents or plasma expanders, for example, that have no biological target. And they may crop up in our hits, and we may not want to pursue some of those molecules. So some knowledge about what the FDA drugs do and the targets they interact with is important. And I'll show you a few examples of things that we thought were promising and things that we thought were not so promising from one of our early projects. So over the summer, like many people, we've looked at a couple of coronavirus proteins. We did some docking work with the SARS-CoV-2 helicase. This is a protein that separates the viral RNA from proteins and duplex RNA or RNA DNA complexes. And it's a potential target because if we can inhibit this enzyme, then it has a potential antiviral effect. And we carried out some docking studies with various sites on this protein and found amongst our hits a range of compounds that looked initially interesting, but after a bit of digging and a bit of knowledge about what some of these drugs do, we were slightly concerned. So I guess remdesivir is a popular drug for repurposing, particularly in relation to coronavirus, largely targeting the RDRP protein, but it cropped up in some docking studies relating to this particular target as well. But we need to be mindful that it's a prodrug and that the form that's administered isn't the bioactive form. So this is a prodrug, it's a protide type drug, phosphoramidite, and it decomposes to produce a monophosphate, and the actual bioactive species is a triphosphate. So it's not an ideal starting point for drug design or as a docked species because it's not the bioactive species, it's a prodrug. And a couple of other molecules that cropped up. So digoxin is a cardiac glycoside. It is very potent, has a low dose and a narrow therapeutic window, so not necessarily suitable for a repurposing project because of that potential for toxicity. Propranolol, I showed you earlier, is a beta blocker, again has potential for lowering patients' blood pressure if they're given it for another purpose. And dinoprostone is an oxytocic agent. It's used to induce labor and as to induce abortions as well. So again, this is a drug which has potential toxicity and isn't necessarily suitable as an antiviral drug. And a few things that cropped up that weren't particularly targeted were things like sucrose. It has a lot of potential to form hydrogen bonds, so it interacted with some polar sites on the protein. But again, we wouldn't even consider this as a potential lead compound because it's polar, it's a food. You have lots of it already. If sucrose cured coronavirus, then lots of people in the Western world would never catch it. And this iovasol is a a radio contrast agent. It has 3 iodines, so it's administered. It stays largely in the plasma, so it's used to stain blood vessels, so you can see occlusions in tissues and measure perfusion and such like. So again, it's retained within a compartment within the body, in the blood, and it's not really a suitable medicine for intracellular targets. But there are a few proteins that we were kind of few ligands that we're kind of interested in, because they have potentially different targets to begin with. So tetracyclines are antibiotics, so they don't have a human target. So they are potentially interesting repurposing candidates. Likewise, dexamethasone. Now, dexamethasone has some therapeutic activity against SARS-CoV-2 anyway, or patients who have been infected with it previously. So it's a potential, the anti-inflammatory effect might be useful for patients to some extent, but if it interacted with a viral target, that might be a desirable thing as well. And we pulled out some other antivirals and anti-inflammatory drugs along the way that looked interesting. So we were interested in kind of screening the molecules for their existing therapeutic activity and asking the question whether it would be okay if this drug was repurposed for a viral infection or not. So the next steps are perhaps to do some more simulation. And in the case of the tetracycline antibiotic, I'm going to show you a movie. So let's just watch and see what happens when we run that dynamics for a bit longer. So it looked great to begin with. The drug seemed to form lots of hydrogen bonds and form a neat interaction with the protein. But as you can see, when we ran just 50 nanoseconds of dynamics, the molecule decided it was going to leave towards the last 15 nanoseconds of that and never came back. So the docking was instructive. We could filter out some of our hits, and we could narrow down the list even more by running some dynamics and seeing if some of our complexes were stable. And I guess you've seen examples of that over the last few days. So what would you do next with some of the hits that you've identified from this kind of approach? Well, I guess the next thing to do is an experiment. So you need to test whether your predicted interaction really works with your particular target. And if you're working on, say, a viral target, for example, you need to establish whether there is a difference in affinity for your known target versus the new target. And that could be an important safety consideration. If your molecule binds to a viral target, for example, at a much lower concentration than its therapeutic concentration for some other target, it might be a useful therapeutic window to exploit. So those things need to be considered. But what do we do if we don't have any promising hits from an FDA-approved drug? They're not easy to modify necessarily. Some of them are natural products, some of them are big molecules. So the undertaking for an experimentalist wanting to make new versions of those drugs could be quite large and quite risky. So probably what we would like to do instead, if we don't come up with a good candidate from a repurposing route, is to design a new molecule. And the premise of fragment-based design is that we can start with very small molecules early in the drug design process and grow them to make larger molecules along the way, and retain many of the desirable properties of small molecules, for example, solubility and good distribution and efficiency in their activity. Now, the total chemical space of very small molecules is much smaller than all chemical space, so we can sample it better with smaller libraries of ligands. And these ligands can potentially interact in an efficient way, so they have fewer obstacles, fewer steric clashes to forming interactions, but generally they bind with lower affinity because they're smaller and form fewer interactions in total. So, for example, from a high throughput screening approach, we might develop a molecule that looks like this. So the structure is shown in yellow and the protein is shown in blue and orange, and the drug extends across the binding site but doesn't form necessarily good interactions with all of the potential sites in that binding cavity. But a group of fragment compounds may, so they may bind separately to each of those sites and bind quite well. But because they're small, they bind with low overall or absolute affinity. And the goal then is to take a fragment hit and grow it into some of these other sites or link them together to produce a new, more active ligand. And we can do this experimentally, and also we can use it as a potential molecular modeling approach as well. So as I said, fragments are typically small. They have a molecular weight, generally speaking, of less than 300, as calculated lipophilicity or log P of less than or equal to 3, fewer than 3 hydrogen bonds, or fewer or equal to, and same for hydrogen bond acceptors. So they're small molecules. And there are many approaches to designing these fragment libraries, including 3D features, for example, and optimizing the size of the compound library. And from a docking perspective, I guess the attractive thing for us is that the procedures are quite fast because the fragments are small, they dock quickly, they have fewer rotatable bonds, so there's less conformational freedom, and we have many to choose from. But fragments interact slightly differently with proteins than larger molecules. So as a whole, this is data from across all or a very large set of protein ligand complexes in the PDB. So you can see that hydrophobic interactions are really important to ligand protein binding, and hydrogen bond interactions too. And then you have lesser effects from other kinds of contacts that are formed in the protein. But the situation with fragments is slightly different. So the distribution is slightly skewed. This is 1,500 drugs chosen at random and 1,500 fragments chosen at random. And you can see that fragments are slightly richer in hydrogen bond interactions than drugs as a whole. Likewise, there are more pi stacking interactions. And it seems that there is a tendency to form more interactions and perhaps some more polar interactions in the form of hydrogen bonds. This is a nice article from MedChem Communications a couple of years ago. And what we'd like to see in our fragment hits, be they virtual or from experiment, is we would like our fragments to be efficient, so have a high binding affinity per non-hydrogen atom. So that means that each atom in the ligand is contributing a significant amount to the binding energy. We'd like to see some hydrogen bonds because those are features of fragment crystal structures, which obviously bind to the protein. We would like there to be accessible functional groups to modify on the ligand. We'd like them to be purchasable or easily synthesized, and we'd like several potential points at which we could modify the structure so that we can grow our fragment and expand our ligand into free space within the protein. All of those features are important. So here's an example of how well this approach can work from a crystallography study. So this is the KEAP1 protein that you saw on Monday. And it's a potential anti-inflammatory drug target, various biological activities that can be exploited for therapeutic use. And this is some work that was done by Aztecs in 2016. So they carried out a high throughput crystallography study, docked a series of small molecule fragments into the binding site of KEAP1, which is kind of at the middle of this beta propeller structure here. And here's one of the fragments in a top-down view on this slide. And they identified a few potential sites at which fragments could bind. So here we have a bicyclic molecule binding in the top of the protein, at the top kind of left-hand side, and a second molecule, this time a sulfonamide type structure on the right-hand side. And this is a phenyl propionic acid kind of binding in the middle of the protein. So 3 fragments, they all bind quite poorly. So their IC50s are in the sort of micromolar range, millimolar range. So they're not very active, but they're small and modifiable. So you can imagine linking a couple of these fragments together, or perhaps all 3 of them. And that's the approach that Aztecs took when they were developing these ligands. So they adopted a kind of fragment linking approach. So first of all, they joined the one in the middle and the one on the left-hand side together. So this produced a molecule that bound pretty much in the same orientation as before. That's not always the case. And it has a much higher affinity, 61 micromolar, still not very active, but much better than where we started. And then they introduced some functionality which was similar to the sulfonamide that we had before. And again, the potency improves quite a lot here, about 20-fold. And a few more changes gave them a sub-micromolar inhibitor of this target. So it's a fairly rapid route through a development process using information from fragment positioning within the binding pocket and fairly rapidly coming up with sub-micromolar inhibitors of this particular protein. It's a really nice study and an example of the approach, and it's an example of where it works, and I'm sure there are many cases where it doesn't. And again, we pursued this kind of approach for our NSP13 helicase target and carried out some docking studies with fragments this time, so much smaller molecules than the FDA drugs, and found some interesting structures. We kind of filtered them based on those criteria that I mentioned before. So we tried to pick out compounds that had good binding energy and compounds that formed at least 1 hydrogen bond to the protein in its docked conformation. And we also tried to pick out fragments that were fairly efficient. So we had a ligand efficiency cutoff, which was based on dividing the docking score by the number of non-hydrogen atoms. So we used those kind of 3 criteria to select fragments from a set of about 2,000 and reduce that to around about 50 to 100. And we can then go through those hits that we have and think about how we might perhaps modify those molecules or merge them, or look for similar molecules that are out there in the zinc database that we could dock and perhaps buy. So here's an example of one of the small molecule fragments that bound to the protein. It's a sulfonamide again, and it overlays with some of the other hits in the group that we selected. So for example, we could consider replacing this carbonyl group with an amine, or replacing a hydrogen bond acceptor with a donor at this position. And a different kind of overlay suggests that we might consider making a molecule with a 6-6 fused ring system rather than a 6-5 fused ring system. So it gives us a few things to think about maximizing the interactions that these molecules make and how we can kind of expand our molecule to occupy more space by combining features of interesting fragments. And very recently, there's been some work done with fragment crystallization in this protein, and some of the hit molecules that you see in those fragment-based screens, there are about 50 of these complexes in the PDB now, are quite similar to some of the docked structures that we have in our data set that we acquired fairly recently. So that's kind of encouraging. Of course, if you go too far down this route, if you continue to keep growing your ligand, you risk making it very complicated, and you risk introducing chemistry which might not be simple and might take a very long time. And you need to be mindful that you need to be in a regime where you're making or designing molecules that could be either purchased or made fairly simply at this stage, because we're still in the sort of hypothesis proving part of a project. So what could you do next? Well, I've kind of alluded to that. You can expand your in silico study and focus on active compounds. You can search for structures in databases which have similar features. For example, you can look in the zinc database and you can use the zinc pharma functionality to represent your molecule as a series of pharmacophores. In this case, I've shown propranolol. So we've got two aryl groups, we've got a hydrogen bond acceptor, we've got a hydrogen bond donor here, and we can look for similar molecules to that within the database and pull out, in this case, about 8,000 compounds that have those kind of core, or some of those core pharmacophore features similar to propranolol. There are also some automated approaches to growing ligands. Sprout is a commercial piece of software, Autogrow non-commercial. And there's a bit of a barrier to entry to some of these software programs, but they can be kind of useful for designing new ligands. But I guess really what we need to do at some point is step out of the virtual world and into the experimental world and do some medicinal chemistry and explore the structure-activity relationships of our ligands and try and refine their target activity, improve the way in which they bind to a protein by doing experiments and trying to prove our virtual studies correct or otherwise. And we need to tune their physicochemical properties. So in this cycle of going from a gene to a drug, we're kind of focusing on this top part where we're generating lead molecules, profiling them and optimizing them. And of course, what needs to follow on from those studies are some preclinical studies and clinical studies before we get to a drug. So those next steps often involve some form of in vitro testing, cell-based assays, for example, determining physicochemical properties. So we're interested in solubility, represented by this log S parameter, log P, an actual experimental value, or log D, a distribution coefficient, physiological pH, which takes into account molecular ionization. We may need some in vivo studies to test efficacy and tolerability and toxicity. ADME relates to absorption, distribution, metabolism, and elimination, and we need to determine those properties for potential drugs that are administered systemically. We need to check them for toxicity. This HERG target relates to cardiac toxicity. We need to understand how they're metabolized by cytochrome P450s and their stability in microsomes. And they need to be formulated, so drugs need to be sufficiently soluble to be injected, for example, or suitable for formulation in tablets and sufficiently stable. And then after that, there are clinical trials. But I guess right at the beginning of the process, we can think about smoothing the way for some of these studies by retaining functionality that keeps a molecule soluble and helps with its partition coefficient, and also by avoiding functional groups in the molecules we develop and the hits that we select in order to avoid toxicity. So we can avoid toxicophores in molecules, for example, nitro groups or Michael acceptors, which might interact covalently with targets or form reactive metabolites, which might cause liver damage, for example. Okay, so a few things to consider. Modeling's great, of course, and it's really important at the moment because a lot of us are stuck at home and don't really have great access to our labs. So we've been doing a lot more of this kind of thing, all of us. We need to be mindful that we're using docking, molecular docking of small molecule ligands as a predictive tool. So we are often interested in new targets where there's no validation for our docking approach or our results. So we're using it as a way to enrich our selection of hit molecules that we could do experiments with. And we're using it as a hypothesis generating activity. We need to test our ideas at an early stage in experiments to check we're heading in the right direction. And we need to think about how feasible those experiments are. Can we get some quick wins by doing some docking, perhaps buying some small molecules to test, or making some simple ones and seeing if our initial design premise and our docking results are validated? And we need to do the right experiments. So for example, if we design a drug to interact with a protein target, it would be nice to do a specific binding assay that looked at binding of the molecule to that protein, rather than, for example, a cell-based assay in which we're looking at, say, cytotoxicity for a cancer drug, where there may be effects from other factors in that complicated environment. So a drug that you design for, say, a cancer target might kill a cancer cell, but it might not be interacting with the target you expect. So you may think your point has been proven, but you may be wrong. Likewise, if your molecule is inactive in a cell, it may well bind very well to your particular protein target, but not enter the cell or be metabolized quickly or be eliminated very quickly. So you'd be right, but think you were wrong. So the right kind of experiment is really important. Now, from discussing some of these points with people in the pharmaceutical industry, I think the opinion of molecular modeling can be a little bit of a roller coaster. So, I think medicinal chemists and pharmacologists will regard it as valuable, or do regard it as valuable, if it's proven to be helpful in drug design. It's possible they might become suspicious, especially if they're cynical people, if it leads them in the wrong direction and we propose things that don't work and lab work is wasted. So I think it's important to manage people's expectations of what they can expect from molecular modeling and docking in particular. I say that as someone who makes things myself and is regularly disappointed. But I think it's fair to say that a careful and qualified use of docking studies in drug design is extremely valuable. And it's a tool that we should use carefully and understand the caveats and potential downsides, but also appreciate the value that it can give us and how it can streamline some of the development process. And of course, if you disagree with me, then you simply need to remember my second or third slide and ask another medicinal chemist for an opinion, and it will be different. Okay, so I think I'll stop there. I've covered most of the things I wanted to say, so I'd be happy to take any questions if people have any questions. Thank you.